Pillalu, dear students, in the previous video we started the sex organs of bryophytes. No? Now in this video we are discussing about the sex organs of the bryophytes in detail. So the sex organs in bryophytes are always multicellular. Sex organs in bryophytes are always multicellular. Always multicellular. And these are jacketed. What is the jacket? The protective covering around the sex organs. Jacketed sex organs you can see. And stalked sex organs. We haven't seen this stalk for the sex organs in the case of next one, that is pteridophytes or gymnosperms or gymnosperms. We don't see. Only in bryophytes we are seeing stalk for the sex organs. Remember that. And here the male sex organ. What is the male sex organ name here? Antheridium. How is this antheridium? This is the antheridium structure. Look here. So this is the stalk. This is the stalk, multicellular stalk. Okay. And this is the antheridial wall. And inside that you are seeing androcyte mother cells. These are all androcyte mother cells. All these androcyte mother cells, they undergo mitosis and they form the male gametes or antherozoids or sperms. We can call these sperms as male gametes or antherozoids. See the structure. This is having two flagella, biflagellated. The antherozoids are biflagellated. With the help of these flagella, these two flagella, they move towards the female sex organ, that is the archegonium. Archegonium. With the help of the water, dehiscence of the antheridium takes place. All these male gametes, they come out through this ostiole or hole and they move towards the archegonium, the female sex organ. Okay. So this is about the male sex organ, that is antheridium. And come to this female sex organ. First time we are seeing this archegonium. First archegoniates, primitive archegoniates. These are the primitive archegoniates, and a gradual reduction in the archegonium also you can see. So in bryophytes, fully developed archegonium we can see. Okay, primitive archegonium means first time we are seeing this archegonium. First archegoniates. So with the stalk, multicellular stalk, what is the shape? Flask shape. Flask shaped with multicellular stalk. And so this is the stalk, multicellular stalk. This is the stalk. And with and a body with venter. This is the venter. This is the venter, bulbous venter. And this is the neck, long neck. Neck. So here in bryophytes you can see the stalk, venter and neck. Stalk, venter and neck. But in pteridophytes, reduction of the archegonium you can see. So there is no stalk, only venter and neck you can see. Venter and neck only in pteridophytes. But what about in gymnosperms? In gymnosperms, there is no stalk, there is no neck. Neck is absent, stalk is also absent. Only venter is present. And next one is angiosperms. In angiosperms, complete archegonium only absent. So in the gradual reduction of the archegonium you can see. So the female sex organ in bryophytes is the archegonium. These are the first archegoniates, primitive archegoniates. Shape is flask and it consists of a multicellular stalk, bulbous venter. This is the venter. This part is the venter. And neck. Neck This is the neck. And when you see this one at the terminal part of this neck, you can see the cover cells also. Cover cells. See the venter. In the venter, you can see the cells. One is oosphere. What is oosphere? That is nothing but female gamete, that is egg. This is the egg cell. Egg cell. Look here, egg cell. And above that, this is the ventral canal cell. Ventral canal cell. This is the ventral canal cell. It consists of oosphere or egg, and a venter canal cell. And you see this neck. In the neck, you can see the neck cells. These are all neck cells. In the neck, we can see the neck canal cells. How many? 6 too many. Look here. This is one neck canal cell, another neck canal cell, another neck canal cell. Like this, 6 too many neck canal cells are present. Here these are the cover cells. How many cover cells are present? Cover cells 4 in number. Cover cells 4 in number. Okay. Once we'll see about this archegonium again. Archegonium is the female sex organ. It is a primitive archegonium. First time we are seeing this. First archegoniates. Bryophytes are the first archegoniates. The shape is flask shape, and it consists of a multicellular stalk, venter, and a neck. Okay. So gradual reduction in the archegonium we can see. In the case of pteridophytes, we can see only venter and neck. Venter and neck. In the case of gymnosperms, no stalk, no neck, only venter is present. In the case of angiosperms, there is no archegonium at all. Okay. So inside the venter of the archegonium, we can see the egg cell. This is the egg cell, and that is called as oosphere or female gamete. Above that, we are seeing the ventral canal cell. Ventral canal cell. And in the neck, we are seeing neck canal cells. They may be 6 to many. And the cover cells are 4 in number. 4 in number. And one more thing, jacket is present, I said. So, venter as well as neck are covered by single layered jacket. Venter, single layered jacket you can see in the venter as well as for the neck. For the neck as well as for the venter, we are seeing single layered jacket. Okay, during maturity, what happens? This venter canal cell and neck canal cells, all these they disintegrate. They disintegrate and they form a mucilaginous substance. So when the mucilaginous substance, when it is formed, the cover cells they separate. Okay, giving the way for the mucilase. It oozes out, and this mucilaginous substance consists of chemical constituents. Okay, and these biflagellated antherozoids, they are attracted towards this chemical constituents which are present in the mucilaginous substance, and with the help of water they swim towards the archegonium. So with the help of, because of these chemical constituents, they are attracted. This type of movement is called as chemotaxy. Chemotaxy. So the male gametes, the antherozoids, they move towards the archegonium. They attract towards the archegonium because of these chemical substances which are present in this mucilaginous substance, which is formed by the degeneration of the venter canal cell and neck canal cells. Remember, venter canal cell, neck canal cells, they degenerate and they form mucilaginous substance that consists of chemical constituents. Due to that chemical constituents, these antherozoids they attract towards the archegonium. And how they move towards the archegonium? With the help of the flagella, they swim in the water, because those are always, they grow in the moist shady places only because of the absence of the vasculature. Okay. So, due to these chemical substances, those are moving towards this. This is called as chemotaxy. With the help of the flagella, they are moving. That's why this type of fertilization means they enter into the neck. These anthrozoids enter into the neck, reaches the venter and fuses with the egg. Fuses with the egg. This is called as fertilization. In the venter of the archegonium, you are seeing the fertilization. Okay. This is called as fertilization. Where is this taking place? Fertilization is taking place in the venter of the archegonium. Venter of the archegonium. Venter of the archegonium. So, this is called as oogamous type of fertilization, because the female gamete is stationary and the male gamete is moving towards this. Okay. And with the help of the flagella, they are moving, no? That's why this type of fertilization is pseudogamous. In angiosperms and in higher gymnosperms, you can see siphonogamous type, no? So, pseudogamous means with the help of the motile organs, these male gametes, they move towards the female gamete. Okay. So, during the development, see what happens, the male gamete and female gamete, male gamete and female gamete, they fuse. As a result of this fusion, what is the result? Zygote. Zygote. The zygote is diploid and the zygote undergoes what? In algae and fungi, zygote is undergoing meiosis, so that there is no embryo at all. These are the first embryophytes, already I said. So, the zygote develops into embryo. How? Through mitotic divisions. So, zygote is undergoing mitosis and it forms the embryo. Okay. First embryophytes. So, this embryo develops into sporophyte. Embryo develops into sporophyte. While it is developing inside this venter, where is the sporophyte is developing? Inside the venter only. Inside the venter. The jacket is there, no? Single layered jacket. So, it breaks when the sporophyte is developing inside. The jacket, it breaks and it comes like a cap above the sporophyte. We call this as calyptra. Calyptra. Okay. Above the sporophyte, we can see that jacket like a venter, calyptra. Okay. So, embryo develops into sporophyte. Now, we are going to see about the sporophyte structure. Sporophyte. Sporophyte. What are the parts of the sporophyte? Parts of the sporophyte. Food, seta and capsule. See, bryophytes are having three classes. We know that. So, what are those three classes? Hepaticopsida means liverworts. Then Anthoceratopsida. Then Bryopsida. But in our NCERT, there is no Anthoceratopsida. Just they have given only Hepaticopsida and Bryopsida. Okay. That's why we are seeing only Hepaticopsida and Anthoceratopsida. Okay. So, the sporophyte. Now, the sporophyte ploidy, this is the result of the fertilization only. Male and female gametes which are haploid, they fuse and they form the diploid zygote. The zygote undergoes mitosis and it forms diploid embryo. Embryo, it develops into sporophyte. That's why this is diploid, remember. So, the sporophyte of these bryophytes consists of food, seta and capsule. But in Hepaticopsida, one of the example is in liverworts. Hepaticopsida or liverworts, both are same. Common name for Hepaticopsida, liverworts. In liverworts, one of the example is Rixia. This is primitive one. It consists of only capsule. Only capsule. Only capsule. But what about in Markensia? That is also one of the liverwort. Markensia. And moss means Bryopsida consists of that mosses. Funaria is one of the example. Funaria. In these two cases, we can see foot, seta, and a capsule. Markensia also a liverwort, but it consists of foot, seta, and capsule. But Rixia is a liverwort, but with only capsule. Only capsule. So, the sporophyte is complete parasite or partial parasite. Where do you see complete parasite and partial parasite? Complete parasite in the case of Markensia. Markensia, sporophyte completely depending on the Markensia, completely depending on the gametophyte. The sporophyte is completely depending. So, complete parasite. Okay. But in Funaria and Anthoceratopsida also, we don't have that Anthoceratopsida. That's why I'm not mentioning that. So, in Bryopsida, mosses, partial parasite, because those are photosynthetic. Partial parasite. So, Markensia is non-photosynthetic. Non-photosynthetic. That's why complete parasite. Moss is partial parasite because those are photosynthetic. Means the sporophyte is having some green cells. Chlorenchyma you can see. Photosynthetic. Okay. Once we see this is Markensia sporophyte. This one. Markensia sporophyte. It is hanging, drooping. And you see this one. This is Anthoceratopsida, Anthoceros sporophyte. And you see this one. This is the sporophyte of the mosses. Foot you can't see. That is embedded inside the gametophyte. Sporophyte parts are foot, seta, and capsule I said. Okay. Foot is embedded inside the gametophyte for getting food. And this is the seta, long and stalked, slender one. And this one is the capsule. Again, the capsule is differentiated into apophysis. This is the apophysis region, and this is theca region, and this is the operculum or lid. Okay. Capsule again it is differentiated into apophysis. We do no need of all this. Apophysis, theca, and operculum or lid. Okay. So, inside this theca is the fertile region. Why? Here you see this is the spore sac. Look here. This is the spore sac. Inside that only this is the spore sac. Inside that only you can see the spore mother cells. These spore mother cells, they undergo, what is the ploidy of the spore mother cells? Diploid. Spore mother cells are diploid. They undergo meiosis and they form the spores. And in the operculum, this is the operculum. In the operculum you can see peristomial teeth in 2 rows. Those are hygroscopic. Okay. Here those are the thin cells present in the operculum. When they absorb water, those are hygroscopic, peristomial teeth. So, here it dehisces and it comes back like a cap and exposing the inner part. So, all these spores they come outside. they come outside. They fall on the substratum and they develop into gametophyte. Spore develops into gametophyte. All these spores are homosporous. Very important. All the spores are homosporous. All the spores are homosporous. All the spores are homosporous means all are of the similar type. All are similar type. But in the case of gymnosperms and angiosperms, there heterosporous condition, microspores, megaspores. But in gymnosperm, pteridophytes, few are homosporous and few are heterosporous. But in bryophytes, all are homosporous only. Important one, homosporous. The spores are homosporous. How these spores are formed? Reduction division in the spore mother cells. This is called as sporic or intermediate meiosis, because in the spore mother cells the meiosis is occurring. That's why sporic or intermediate meiosis. In the zygote, if it is taking place, means zygotic meiosis. We have seen that in the algae as well as in fungi. Intermediate meiosis. Okay. And how these spores are liberated out? How the spores are liberated out? You see this one in Markensia, you see. See these small, long structures, slender structures you are seeing now. Slender structures. These slender like this. These slender structures. These slender structures are called like these tapered ends with the spiral thickenings. Double spiral thickenings. Double spiral thickenings you can see here in the elatus, unicellular only like this. Okay, spiral thickenings you can see. Double spiral thickenings. These are called as elatus. Elatus. In the case of Markensia, the spores are liberated out with the help of the elatus. What are these elatus? Some of the spore mother cells, they become sterile. Spore mother cells. Sterile. They don't undergo meiosis. These are diploid, you know. So, they develop into elatus. So, elatus also diploid, remember. Elatus also diploid. Unicellular. Elatus are unicellular and diploid. They develop from the sterile spore mother cells. And what about in Funaria or mosses? Peristomial teeth. In Funaria, peristomial teeth. With the help of the peristomial teeth, the spores are liberated. With the help of the peristomial teeth, the spores are liberated out. Okay, and a spore develops into, it germinates and it develops into a gametophyte. Develops into a gametophyte. What is the life cycle here? Which one is the dominant phase? Gametophyte. Gametophyte ploid is haploid. So, haplo, on that the sporophyte, which is diploid, is depending. That's why haplo diplontic. Diplontic. And it shows heteromorphic alternation of generations. How? The gametophyte produces sporophyte. Sporophyte produces gametophyte. So that is heteromorphic alternation of generations. Heteromorphic alternation of generations. You can see here. Okay. So, in this video, we have described or discussed about the sex organs, fertilization and the sporophyte. And in the next video, we discuss about the classes What are those two classes? Hepatic Apsida and Bryopsida. Okay, students, bye.